Plant breeding methods. Plant breeding methods are the approaches used for genetic improvement of plants. Plant breeding methods are separately categorized for self-pollinated crops, cross-pollinated crops, and asexually propagated crops. Self-pollinated crops are also called as autogamous crops, and cross-pollinated crops are also called as allogamous crops. The various breeding methods for self-pollinated crops are as follows: introduction, selection, selection, which is again categorized as pure line selection and mass selection. Pure line selection is the one which is maximum used in case of self-pollinated crops, whereas mass selection is minimally used in case of self-pollinated crops. And the next breeding methods are hybridization, followed by pedigree method of selection, bulk method of selection, single seed descent method of selection, backcross method of selection, polyploidy breeding, mutation breeding, transgenic breeding, and heterosis breeding. These are the various breeding methods for self-pollinated crops. And the next is the breeding methods for cross-pollinated crops. Cross-pollinated crops are also called as allogamous crops. And the various breeding methods adopted for cross-pollinated crops are introduction, selection, again divided as pure line selection and mass selection. In contrast to self-pollinated crops, in cross-pollinated crops, pure line selection is minimally used, whereas mass selection is used in large scale. Whereas in self-pollinated crops, mass selection is minimally used and pure line selection is used on a large scale. And the next approach is hybridization, which is followed by pedigree method for development of inbred lines, then recurrent selection, which again has simple recurrent selection, specific combining ability, and general combining ability, then mutation breeding. Then the next breeding methods for cross-pollinated crops are polyploidy breeding, transgenic breeding, and finally the most important, synthetics and composites. These are the various breeding methods adopted for cross-pollinated crops. And the next one is the breeding methods adopted for asexually propagated crops. The various breeding methods adopted for asexually propagated crops are introduction, clonal selection. A clone is nothing but an individual developed from asexual reproduction. And the next breeding method is hybridization, polyploidy breeding, mutation breeding, and transgenic breeding. And now we'll just have an outlook of the breeding population. Breeding population has homogeneous population or heterogeneous population. A homogeneous population and heterogeneous population may have homozygous individuals or heterozygous individuals. So, first let's know about the homozygous individuals and heterozygous individuals. Homozygous individuals are those which have same genetic constitution. That is, the genetic background of homozygous individuals have same alleles. For example, if the height is taken into consideration, then that particular trait which is represented by the letter T and the alleles which constitute this character are capital T for tallness and small t for dwarfness. If an individual is said to be homozygous, then it will have the genetic constitution as either capital T or small t. If an individual is said to be heterozygous, then the genetic constitution will be capital T and small t. That is, a heterozygous individual has a different allelic constitution in its genetic background. And now let's see about the individuals which constitute the population. A homogeneous population is nothing but which has all the individuals with similar genetic constitution. That is, that group of individuals which are said to be homogeneous can have either entirely homogeneous, homozygous, tall character or entire population can have homozygous small d character, or the entire population can also have a heterozygous allelic constitution. In case of heterogeneous population, a heterogeneous population is a combination of both homozygous individuals and heterozygous individuals. That is, a particular population is said to be heterogeneous if it has a combination and a mixture of both homozygous individuals and heterozygous individuals. That is, that particular population may have combination of individuals which have capital T genetic constitution, small t genetic constitution, along with capital T small t constitution. Combination of any of these three can be there. And in this topic, we have seen about the various breeding methods for self-pollinated, cross-pollinated, asexually propagated crops, and the breeding population details. In the next lecture, we are going to see about the details of the breeding method introduction. Thank you.